So welcome everybody. As I've done yesterday, I'm going to say good morning, good afternoon, or good day, depending on from where in the world you're tuning in. We've had a really exciting couple of days, and I hope that most of you have been able to also look at our YouTube channel. The link is again in the chat. So this, as well as all the other live events, is recorded and will be posted there as soon as we get to uploading everything, and then will remain there. So there's no great rush to watch this. Just kind of to reiterate, we have almost 40 speakers this year, and the channel really is a fantastic group of different speakers, anything from governmental agencies to researchers, and as we have today, implementation partners as well. So we, as I just said, we're recording today's session. And for those of you that have tuned in, and we're getting a really nice count here, please feel free to ask questions during our session here today. We'll have about an hour. And when you do so, please submit this in the Q&A panel and not in the chat. There, I probably miss it, so the Q&A is a lot easier. And we'll be happy to incorporate this in our little conversation here today. So with that, it is my great pleasure to introduce to you Dr. Bill McLamb, who really has had an incredible road in his career towards his current position as the Director for Science and Technology Management at Space Tango. He started, and maybe he will maybe correct me on some of this, but what I've been gathering as an electron microscopist, then became a clean room engineer, which looked to me was his first job at KSC, so at the Kennedy Space Center. Then he was a mission operations scientist and a payload development engineer also at KSC. He was then a research scientist at Dynamac Corporations, where they also did a lot of space-based research and space biology. He obtained a master's degree and a PhD from the Florida Institute of Technology and was funded both by the NSF and the National Defense Science and Engineering Graduate Fellowship. He then went on as a researcher for the University of Central Florida at the Nanoscience Technology Center, and later joined CASES and the ISS National Labs as their operations manager. And now, I believe for roughly a year, just a little bit shorter than a year, he is with Space Tango, and we're really excited that he can join us here today to give us the insides and the outsides of Space Tango, and how, as a researcher, we can, work with this company in order to bring our science into space. So with that, welcome to our discussion here today, Bill, and thank you for your time.
Thank you, Christine. And I suppose I need to share my screen.
Yes, I can stop share. Here you go.
PowerPoint. Now see my screen?
Perfect. Yes.
Okay, and now let me put it in presentation mode. Okay. Well, thank you very much, Christine, for that wonderful introduction, and for the kind invitation to present at this second Australian Space Biology Symposium. It's exciting to watch the evolution of a new Australian space agency, and we certainly look forward to opportunities to work with our future space colleagues in Australia and, of course, anywhere else in the world and off the world. I'm happy to talk about space with you today about Space Tango, and provide some insight into Space Tango's capabilities and partnerships for stem cell discovery and manufacturing applications in microgravity. As Christine mentioned, my background is in biology, so that's where my emphasis has been in the space biology world for some time. In our microgravity pursuits, we strive to provide outcomes that will benefit life on Earth, as a space environment can enable a better understanding of the behavior of stem cells and derived tissues and other cell types when grown under the unique environment of microgravity. And we currently explore these ideas 250 miles up in the microgravity environment of the International Space Station. But the apogee of our Space Tango's future achievements for on-orbit manufacturing will be with Space Tango's autonomous free flyer, the ST-42. I think it's over one of my shoulders. I'm not sure, on the screen behind me. And we look forward to sharing more information about that in the coming months and years as the system gets developed. And we're happy to talk about this some more today during the question and answer session, and my contact information at the end for any other questions you may have. So as I mentioned, our current platform for innovation is still the International Space Station as we use as an incubator for research and development and for pilot manufacturing. From there, we'll look in the future to commercial space stations that extend the current use of the ISS, such as the Axiom Space commercial space station, and excuse me, as an incubating hub for new product development. And then we'll move to the independent spacecraft like the ST-42 for scalable production activities. So at Space Tango, our vision is to use the unique environment of microgravity of space to inspire and innovate and create a better future for humanity. And with a mission to manufacture biomedical and technology products in space that create value and transformational solutions for humanity. So we target opportunities and partnerships that will enable us to explore fundamental science questions, but also look to build a commercial space economy for the development of biomedical products in space. While our innovation platform is 250 miles up, our actual innovators are on the ground and headquartered primarily in Lexington, Kentucky, where our CEO and founder, Bill McLamb, and most of our team reside. Our commercialization hub is in San Diego, where Jenna Studemeyer is our commercial innovation officer. I think she may be on the call today. So if you have specific questions about commercialization, I will encourage her to speak up. We can also, during the chat session or after for future discussions down the road. We have a strategic engagement office where Cindy Martin Brennan is near Washington, DC. Our launch operations primarily at Kennedy Space Center and Wallops Flight Facility. Those are the primary launch facilities we operate from now. I'm at Kennedy Space Center and have been for pretty much my entire life. I grew up here, so I'm a child of the space program, and it never gets old for me here. The actual facilities that we operate on the International Space Station, the Tango Lab and the PAL, the Payload Ascent Utility Locker, I'll speak about a little bit more in just a few minutes. So, and those two platforms that we do perform our research in were designed to be cross-compatible. So they can integrate and operate within the current and future launch vehicles and future space facilities. And so some of you may be familiar with the vehicles that are listed here. Current commercial resupply launch providers like the SpaceX's Falcon 9 and the Dragon capsule, and Northrop Grumman's Antares rocket and Cygnus capsule. The Falcon 9 and the Dragon capsule, the Dragon capsule docks to the space station and can transport cargo and supplies to the station. It can also return. Now it used to splash down in California, is now scheduled to splash down off the coast of Florida, East Coast, or in the Gulf of Mexico. So there is a return capability here. The Northrop Grumman system, the Cygnus capsule, is not designed for return. So it is a transport up capability. So if you send something up in space on a Northrop Grumman vehicle, you'll wait for a ride back on the Dragon capsule coming down. So a little bit more about the NASA commercial launch providers. The NASA Commercial Crew Program with SpaceX and Boeing, upcoming private astronaut missions with Axiom Space and SpaceX, and Sierra Nevada's Dream Chaser, which is shown here in the coming years. As I mentioned before, we are looking to operate on future commercial space facilities to maintain a permanent presence in low Earth orbit and on facilities used by Axiom Space and also our own Space Tango ST-42, which someday may find a more attractive name, but for now, that's still what we call it. So some of you that may be new to space biology may be wondering, how do I translate my ground science to a space research project? What are the challenges? What are the considerations to ensure that that it will work? I've always been told and taught colleagues that you essentially have to make it bulletproof. Robust experiment, both in experiment design and payload hardware design to support an successful experiment, because of the rigors of space flight, the things that are still a challenge today that we've been dealing with throughout at least my career in space biology. Some of the things that we still deal with, we'll always deal with, because the rockets are of a limited size and capacity. You have limited power for your experiment if it is a powered facility or device, both on the rocket during ascent, once on the space station. You have limitations on volume, weight, and CG during launch and return operations. Obviously, the vibrations of launch and the G-forces from launch and splashdown are things to be concerned about if you have a delicate cell culture system that is only mildly adherent to the surface, for instance, and you shake it up, it's not going to be very happy. So, and these are all parts of the process for development and validation of your plan, to make sure that once you get it to space flight, all the preliminary testing that's done can minimize the risks to success. Other things that we have to worry about is how do you operate your experiment? A lot of what I'll talk about today is how Space Tango uses automation as a primary focus for operating our payloads. So how do we command those facilities, and when and how can we command them? There are some limitations when you go up on orbit. On ascent, you have very limited capabilities for command and control. You can monitor, but not necessarily drive a lot of functions during that time period. Safety hazards and controls are primary, the number one concern, in my opinion, and always have been, particularly when you're dealing with manned space flight. You want to make sure whatever you put in that manned vehicle, in that habitable volume, it's safe to the crew and to the vehicle. And there's a very robust process in NASA's space program to ensure that that all those requirements are met. Because of the unique nature of microgravity, the fluids and gases just are you have to deal with different issues with them, because the lack of sedimentation and buoyancy-driven convection, they just don't perform the same as they would on the ground. That's where we have issues. Often have issues with bubbles, particularly in microfluidic systems that we use in our cell culture devices. That is a constant concern and problem that needs specific corrective actions. As far as how you collect or how you collect your samples and analyze your samples, some opportunities for on-orbit operation allow you to collect samples manually. You have crew-tended operations where the crew can perform certain activities. Obviously, these experiments and these payloads that we fly are not their only responsibilities. They're busy taking care of the place in which they live and work. So crew time is not, it is a luxury. There's always available time. It has to be scheduled accordingly. So one of the reasons we go with automated systems is it gives us the freedom to perform those operations, design our experiments in such a way that we can perform, we can operate as needed. For those, for live cell culture experiments or other experiments that require conditioned storage, like preservation with chemical fixation, or with perhaps you're preserving your RNA with something like RNA later, which is routinely used on the space station, or other types of preservation solutions. And refrigeration on orbit is a limited capability. It's something that is always in demand particularly for any live science experiments. So how you design these experiments and how you package them, how you operate them, how you collect them in return has to be well thought out. And one thing that as far as life sciences goes, contamination obviously always a problem. So something that you have to address and make sure that your hardware systems, your fluidic systems are well designed to minimize that risk. So Space Tango prides itself as a leader in an automated system that support iterative R&D and manufacturing on ISS. And the automation provides us with many benefits. It provides an opportunity for greater sample numbers that and yielding a higher throughput and improved consistency. The cell culture platforms that we have developed are equipped with sensors and controls for temperature and pressure and flow, imaging systems, and these automated systems and this known performance help us to standardize the science environment. So you have a system that's of repeatability. The automation also allows us for on-orbit evaluation of our subsystems and technologies that will require, that will eventually be required for future fully integrated flight experiments. So we may choose to fly subsystems like our fluidic system or our imaging system on a validation flight without biology. Because we are frequently flying we have opportunities for frequent access to the space station. We have facilities that reside on there permanently that we can do this iterative testing to allow us to better prepare the system. So when we do fully integrate them for biology experiments, we've mitigated some of those risks with these new technologies. We also have a step-by-step development process that provides this early validation and extensive early and iterative ground testing that ensures that the system designs will meet the science needs. And as a biologist and as a scientist, working with engineers all my life and working in the area of space biology, and also with Space Tango, our primary focus is always, what are the science requirements? We want to meet those requirements. So that's something that is first and foremost at the beginning of our any development process, any product development. We start with, what are the science needs? So I'm sure scientists in the audience will be happy to hear that. We have data management capabilities with our on-orbit facilities that enables transmission to our Space Tango ground stations. And that allows us to make adjustments and changes in real time, which may not be possible for some of the, for manually operated systems. And our automated devices also provide a reproducible environment to ensure that the validity of the ground control experiments. So we demonstrate this focus on automation with a couple of the facilities that are now on Space Station. The Tango Lab, which has been there for a few years now, which provides a fully automated system for data management, data handling, and telemetry, and simultaneous independent operation of multiple payloads within a Tango Lab system. And the Powered Ascent Utility Locker, the PAL, which, as the name defines it, is used for powered ascent of payloads that are loaded into this locker. So it provides ascent capabilities for with power that can provide for, in the case of cell cultures, for media exchange, temperature and gas control, and data collection. As I mentioned before, we don't have a whole lot of capabilities for commanding specific operations like imaging and data downlink, imaging downlink while on ascent. That typically waits till the systems are transferred to docked and transferred to the station and installed for continuous operation. So just to give you a little more information about the systems that are at the hub of what supports the cell culture is our cell culture cube lab. So this system is in the case of the design here is a 9U cube lab. So for those of you that aren't familiar with the cube lab unit structure, that one unit is a 10 centimeter by 10 centimeter cube. And so this 9U system is typical for our operations. We can support 2 of these in a system that's a little bit larger than a microwave oven. They have capabilities for thermal control, for cell culture. We have so the entire system is thermally controlled. They have TECs that reject the heat from inside the box. So there's always the heat generated from the internal systems. And we have this, it looks like a thermos, or what we call our cold flask, is our essentially our refrigerator. It's for thermal management control of media and reagents that we can typically keep at 4 degrees C. We have a fluidic pumping system and manifold system that can process multiple types of fluid, whether it be cell culture medium, chemical fixatives, RNA preservatives, and this is all automated through the Cube Lab flight computer, which is in the bottom of the Cube Lab, and can be operated remotely and automated and by pre-programmed protocols in the system. We do have which is new to our Cube Lab system, is an imaging system. I'll talk about it a little bit more on the following pages. Is a system for bright field, phase contrast, and fluorescent imaging on a 3-axis gantry to allow for full motion to the images, to your imaging of your samples. This rectangular piece here, this is actually a SolidWorks drawing of a payload that's going up on SpaceX 21, Siobhan Mulaney's project from University of Florida, not too far from Mayo Clinic, and she's flying a muscle atrophy study. This manifold has, this is one of the NCAT's tissue chips projects, is a microfluidic system that is fed by the automated fluid manifold, and the imaging system will take images of each individual sample at pre-programmed times. The system is designed with a homing capability, so you can go to the precise point, return to the same point as the protocol requires. The We do have capabilities for live or cold stowage sample return. So the live return, the box itself would be maintained at the desired incubator temperature if you're bringing back live cell cultures, and remain in conditioned stowage through descent, recovery, and return to the PI or partner involved in the experiment, and where you can then do your deintegration and sample processing on the ground. They also use the same capabilities that NASA provides for cold sample return. So if we had to preserve the samples, for instance, in an RNA later preservative, and we wanted to keep them at refrigerated temperatures, and in some cases we would just refrigerate this entire or freeze this entire cube. But because of as I mentioned before, the limitations for cold stowage or any stowage on space station is it's a premium. Then we look to reduce this volume as much as we can, and we certainly don't really want to put our microscopes in the freezer. So we developed capabilities to essentially take the lid off, take the top half of this box off. The microscope, which is hard mounted into the lid of this cube lab, would then remain on the space station. Or we could then integrate it with a bottom cube that has a new experiment that comes up at a later date. So it allows us to reduce our overall cold stowage volume needs, which makes NASA happy, and saves us from having to replace our microscope every time we put it in the freezer. We're also looking at other ways. We can certainly for media samples or for conditioned media collection, or samples of fluid from your cultures, they can be pumped back into the cold flask, into sample bags, and maintained at the desired temperature till return. We're also looking into capabilities for removal of samples or cartridges from the Cube Lab as part of future Cube Lab designs. It takes a little bit more crew time and on orbit, but we attempt to try to meet the scientists' needs, and the need for access to samples, particularly longitudinal samples, during the course of an experiment is always something that is requested. In some cases, we just may have initiate the experiment on orbit and terminate it at the endpoint, collecting automated data and not removing samples. So there's different. It just depends on the experiment design. As far as sample configurations, I mentioned the manifold system. It's a metal manifold that provides a thermal stability, but also machined into the system, into the manifold, are fluidic channels that can feed multiple locations. The designs themselves, the fluid mapping, the fluid architecture within the manifold can be experiment specific, so it's not a set design. We can also, the shape of the culture chambers themselves can change based on user needs. We also can provide systems that use cell culture bags or multi-well plates. I've already mentioned the tissue chips that are illustrated here. And if the user or partner has a specific experiment unique need that we don't already have developed, upon validation of a system like that on the ground, we could then consider including that. What we're doing with our overall system is developing primary subsystems that can be used for a multitude of experiments. So we have an incubator system, a manifold system for fluidic delivery, a cold flask for refrigeration, and an imaging system. So those are the primary subsystems. And then the specific question is, what do you want in the box? What science do you need to put in there? And that's where those are the early details where we identify what are the science needs and how does it best plug into our overall system. And that's part of the fun, because we get to help help you design an experiment that can help you answer those unique science questions. So a little bit about our microscope system. It's based on a commercially available laboratory incubator microscope, the Etaluma system, that in partnership with Ataluma, we've taken this system and essentially reconfigured it so it would fit into this 9U Cube Lab. So it uses the same optics. It's just bent in different ways so it fits into the lid of our box. It's got the 3-axis motion system, and we have the capabilities for bright field, phase contrast, and fluorescence microscopy. For the fluorescence imaging, Ataluma has a deconvolution software that provides near confocal capabilities. We do have remote benchtop capabilities for support of our experiments as well, both for early ground testing and processing when PIs and partners want to do integrated testing with at least, for instance, the fluidic system and the imaging system, but maybe we don't have the fully integrated Cube Lab yet. It allows them to perform experiments under that emulate how the system would operate in a fully integrated system. So we can provide a fluidic manifold system with a pumping system that uses the same devices. We have the user interfaces that can provide performance data, whether it's pump speed or power or frequency of events. And we have Wi-Fi capabilities that allows us to, so we can provide this to the partner in their laboratory, and then we can monitor and control via Wi-Fi access from Space Tango headquarters. We use the Grafana portal for most of our data posting, so you can monitor this in real time. Whether, is the pump working properly? Are the valves opening as they should? Are the temperatures as they should? I'm monitoring just the exact same thing for an experiment we're doing right now for Dr. Mulaney's experiment that's flying in a couple of weeks. So we eagerly look at this data every day. So it's a nice feature to have. So it's not something you have to wait for someone to report to you, what the data is looking like. We have the same capabilities when we're flying on orbit, monitoring the operations of the Cube Labs and the Paul facility. So it's a user interface that our partners are really happy with. So that's the general capabilities of the systems. I want to just briefly mention some of the programs and partnerships that we have. I do want to save a lot of time for questions and answers. I don't want to run out of time here, but I did want to highlight some of the exciting projects that we have been a part of, and that may give you some ideas of what type of work you may want to do, or at least let you know what's possible to do in space. So in my previous life before Space Tango, I worked with CASIS and the ISS National Laboratory, and we were very fortunate to get involved with the collaboration with NIH NCATS on the the Chips in Space program, something that I think we had while it's all said and done, over 20 flight projects, 20 flights with multiple projects flying. Most of the NCATS projects have two flights. For those of you that are familiar with the NIH funding process, it's a UG3, UH3 funding cycle. So the UG3 phase would support the first flight, and if successful, would move on to the UH3 phase. The NSF in CASIS, again, when I was still there and still continuing, had a collaborative solicitation for tissue engineering and mechanobiology on ISS. I'm a sensory biology guy, so I was pretty excited about this one. For those of you that are interested in something that may be coming up soon, this solicitation was just re-released, I think a couple of weeks ago, and in January, there is a preliminary proposal submission deadline. So I encourage you to look on their website, and if you need a link, please reach out to myself or Jana, and we'd be happy to share that. We've been fortunate to work on quite a few of these projects, and always looking for more opportunities. We're also really excited about recent opportunities with NASA for commercialization focused on regenerative medicine. So just briefly about some of the projects in the Tissue Chips program. While not all of these listed here were supported by Space Tango, there are other hardware providers, implementation partners, as we're now called with the ISS, that can support your experiments. But these are some of the projects that we've been involved with. And the first two with Children's Hospital in Pennsylvania, the Lung Host Defense, and the Blood-Brain Barrier project with Emulate are two that we supported on the very first NIH Tissue Chips in Space solicitation. And then we came on Tissue Chips in Space 2.0. NCATS was so happy with the first solicitation, they immediately released another, and we were lucky. Of the 4 that were submitted, the 4 that were selected, we were part of 2 of those. The second one listed, the Human Myocytes project with Siobhan Mullaney, is the one that we'll be flying in 2 weeks, and the Emulate project with his enteric physiology. It's a gut tissue chip and with sensory neurons. We'll be flying, I believe, on SpaceX 22 later this year. Or I'm sorry, later next year. And as I mentioned, the tissue engineering solicitations with tissue engineering mechanobiology, there have been, I think, 3 solicitations now. And we've, I think, out of the 10 that have been awarded, I think maybe we're involved in at least 6 of those, and happy to report that the most recent solicitation awards that were just came out a month or so ago, 2 of the 3 that were awarded that we're going to be working on. Look to the websites here. You can find out more information about each of these projects, and you can see what type of work is proposed and selected. And as I briefly mentioned, the most recent solicitations with NASA and the awards for commercialization focus in regenerative medicine are really exciting projects that, in this case, unlike the NCATS projects that have 2 flights and the NSF projects that only have 1 flight, some of these projects have multiple flights, 3 to 6 flights over a period of several years. So these are really exciting projects. The first one with Lambda Vision, they're looking at creating a protein-based retinal implant for manufacturing on the International Space Station. We've already conducted a pilot project flight with them. I think it was before I was with Space Tango, a year or so ago, when I was with National Lab. We're excited to be more involved with them again on this partnership. With the UC San Diego and the Stanford Consortium for Regenerative Medicine, we're also working on a stem cell orbital research laboratory for stem cell translational medicine. So it would be a stem cell research lab on the ISS. So that's another exciting project that is just now getting started. And then another project with Cedars-Sinai, where looking at stem cell manufacturing. One of the other types of projects that we've been more involved with recently is with human brain organoid models that both with the UC San Diego and the National Stem Cell Foundation, who have each developed their organoid models for neurodevelopmental biology and neurodegenerative disease studies. The human brain organoid model with UC San Diego is another one we're excited about. They've done some preliminary work on exploratory flight back on SpaceX 18 flight, and this is another project that is getting ready to fly in just a couple of weeks on SpaceX 21. So one of the other projects that we're very excited to see get off the ground. This is you can see from I think their previous flight, the base of the Cube Lab and the media bags and fluidic systems. This is an earlier design where we used to have a lot more of these fluid lines. We've minimized that dish of spaghetti and created this microfluidic manifold system to better manage fluid handling and fluid routing. And this is just a brief image from the National Stem Cell Foundation project. This image is their organoid undergoing microglial migration. And we do have, I believe, a third flight scheduled for this project later next year. So with that, I'd like to say thank you for the opportunity to present today. Really excited to see more space biologists from down under getting involved in space. And I'm happy to take any questions from anyone. And if, as I said, Jana Studemeyer might be on too, if you have questions that are specific to our commercialization efforts. She's been with Space Tango for years now, and her background is well suited to answer those questions. But happy to discuss more.
Fantastic. Thank you, Bill. This was really a great overview, and it's really remarkable, like the capabilities that are already there now, you know, after, I mean, I guess essentially just a few years of really, you know, focusing on space biology. And so it's really fantastic. I am going to take the liberty and make Jana a co-host. So if she would like to, she's welcome to join us, but no pressure. Welcome and thank you. I guess I do have, you know, we have a couple of questions that we were talking about. I think one of the most pressing ones for researchers is, could you give like an approximate timeline on, you know, a researcher would come and contact Space Tango, like from the start of, okay, here's my idea, maybe I don't know exactly what technology I need, how long will it take me to get to flight? I know this is a little bit of a hard one because it really depends on what you're sending, but just kind of to get an idea, what are the milestones?
Yeah, and you're right, it depends on the complexity of the experiment, and I think it also depends on how well defined the protocols you're using need to, and how translatable they are to the new environment. You know, sometimes it just takes a little bit of ground testing to, let's say for instance, you change your media every 2 days in the lab because that's the way you've always done it. Well, we have, you know, if it was a manual experiment, you don't have the opportunity to change that media on ascent for a couple of days, and as you're waiting to dock and transfer and unload and reinstall on ISS, it could be, you know, 3 to 5 days before you can touch your samples again. So, you know, a lot of the payload developers like us, we want to find out what the boundaries on the requirements are. You know, it's a plus or minus 2 days, or 2 days plus or minus 3, or so it's really about finding out what the acceptable limits are for anything, whether it be fluid exchanges, temperature control, you know, how often do you really need to take a picture? If you took a picture, do you need to see it right away? And then if you're developing if we have to develop a device that needs to perform some unique function, that requires iterative testing on the ground first with prototypes. It really does depend on, you know, are you just going to be able to take your device that you already have on the ground, make some modifications and plug it into our subsystems, into our Cube Lab? Maybe you could fly within a year or less. If it takes more involved testing and development and design, maybe 18 months. So, you know, we're a little bit, we target a little bit faster, I think, than some of the other implementation partners, but I think because we're trying to focus on reusing the same subsystems and limiting the changes except when necessary, just to those unique aspects where your biology interfaces with our system. So that we hope that accelerates our process.
Mm-hmm. No, that's very great. Yeah, the focus on the biology is, I think, also very unique to Space Tango versus some of the other implementation partners that have other, also a lot of other aspects in their kind of focus area or focus range. We have a question from the audience. What is the most pressing problem that needs to be attended in order to carry out more diverse experiments related to space biology in space? I think that may be a little bit of a big question. maybe Federico could also give a separate example of some details there. But go ahead, Bill, if you have some thoughts on that.
My first thought was bubbles. If you're talking about life sciences, fluidic systems, bubbles are always an issue. I think that you certainly want to have systems that are reproducible, that are well defined. So every time you operate them, they perform in the same way. The challenge for anyone that's flying something in space, or for any experiment for that matter, is you want to limit your experiment to test that one unique variable, which is microgravity. So, but if you've got a system that, we used to call them hardware effects, or that may not be attributed to microgravity. It may be because the system isn't designed well enough to provide the ideal thermal environment, or airflow or temperature, and those variables in conjunction with microgravity maybe exacerbate a problem that may mask the microgravity change that you're looking for. So I think just having a robust system that can eliminate that variability and allow you to truly test your hypothesis.
Mm-hmm. So you showed the different capabilities, and some of which is obviously brand new with some of the imaging, etc. How modular are the systems when a researcher comes and has maybe a very specific a readout in mind, or a very specific experiment, or something that they want to test? How flexible are you as a company to say, okay, we're going to investigate this and we'll make it happen? Obviously a question of cost, but is this something that is within the realm of capabilities and manpower, I guess?
Yes, and we do have, I think I know Twine, I can speak for Twyman. He likes the unique challenges and do the things that haven't been done before. And if since our systems are essentially, power, data, fluids, optics, we can find ways to integrate into our systems that provide that capability, collect that data, hopefully with minimal change to our systems. What we're trying to get away from is designing a completely new cube lab every time we fly it. So anything we don't have to change, we use the same. And that way every time we fly it, we get more runtime. We increase our comfort with the system, or knowledge of the system, how it really operates. So we don't want to keep changing every time. So, but we like to first just find out what the requirement is and see how best we can meet that. And I think that the NSF projects, the tissue engineering projects, are a good example of that. Some very unique and interesting experiments that that people come up with, and that we just find a way to address the question. And so yeah, we're as flexible as we can be. I don't think there's anything we've turned away yet. That is technically feasible. Yeah, happy to always look for new ways to do things.
Mm-hmm. Mm-hmm. And bar knowing what, what I as a researcher want, how much technical knowledge do I need to bring? Bar, of course, if I knew everything, that would always be great. But, if I'm more comfortable with the biology, is the rest, and I know you kind of touched on this, is that something where, you guys just then, kind of take over and teach me as the researcher, okay, this is what you need, and this is how we're going to change this because we're on orbit and we have, we're changing things to.
Yeah, so I think the screen froze. Can you hear me?
Microfluidic systems or et cetera, like that.
Oh, sorry, the screen froze for a minute, but as far as what do you really need to know and come to us with?
I think we're frozen.
Let's see. Yeah, it's freezing in and out.
Yep, I can hear you.
Okay, so I think it's freezing on both sides back and forth, but I'll try to answer the question, and maybe Jenna's trying to chime in.
I think we're good. Sorry, there was some glitch here.
All right. Okay, great. So what do you really need to come to us with? Honestly, it's just come with your idea. And you don't need to really know much about spaceflight. We want to eliminate that concern and question. And if you have a science question, we help you just to package it in a way that it's feasible to perform. What we don't want to do is change your science in such a way that it's not allowing you to ask that question. So, and I think that's just a challenge in general when the biologists want something and engineers want to design it a certain way. It has to, again, it comes back to what are your needs? What are the requirements? To me, it's always about requirements and capabilities. So what are your science requirements and what capabilities do we need to provide in order to meet those requirements? Allow you to ask that question. And I learned early on in this business that I'm never the smartest one in the room, but I always know where to find the smart people. And if we have a question we can't answer, there's certainly a lot of experts out there. And so come with your ideas, and we'll help you find a solution.
How is the kind of the streamlined process of, the researcher bringing the idea to you building, or adjusting the cube labs, for example, to the needs, and then kind of navigating the way onto the ISS, essentially? Is that something that's also, where Space Tango is also heavily involved, or is this more back to the researcher and the research group?
On how to get to space.
Mm-hmm.
So we, as I mentioned, we have facilities on orbit, and because of that we send our facilities up on the resupply vehicles that are already taking crew and cargo up there. It's certainly the rocket costs are always the most expensive. But not a cost that has typically been incurred by us as users of this resupply chain. So what the costs that are typically associated with are the development costs for the hardware testing, design, build, operate, and fly of the experiment, and then those user costs, the partner costs from your science costs. What would it take to perform the experiments, collect the ground data, the preliminary data to allow you to validate those experiments before you fly, and then all the costs for processing and analysis. So there are different buckets of money that need to be addressed. But we can, if you we can help you get to the as far as the allocation to get to space. Mm-hmm. And Janet, you might want to chime in.
Oh yeah. If there's something else to add, that would be great. Hi, thank you for joining us, Jana.
Hey, thank you very much for adding me in, and I appreciate the opportunity to be speaking with you today. So yeah, in terms of the, getting to space, obviously there's a lot of safety testing that gets done, mission integration, all of that is done by the Space Tango team as well. So, as Bill mentioned, really kind of coming to us just with the idea that you have, and then we'll help you translate that into the flight hardware that can support those science objectives and also get you all the way to station so that you can actually perform your work on orbit.
That's fantastic. Yeah, I think those kind of streamlined processes are very helpful for the scientist that wants to do the science and not necessarily wants to spend their entire life trying to figure out how to find a spot on a rocket, right?
Yeah,
Bar for
exactly.
themselves, probably.
Well, and the truth is, the science has become much more complicated too than what we were capable of doing. Typically, cell culture is something that has been done on station for a long period of time. Stem cell biology, organoids, even the microphysiologic systems and tissue chips, it's much more sophisticated biology now that we're working with as well. And so, we're excited because as we develop these capabilities that can support that sort of next level of science, but often we find we're working in areas where the science itself, still on the ground, is developed. And so it can be challenging, to figure out how to translate that developing science into automated flight hardware and then put it up on station. But, we recognize that it's driving our accelerated understanding in terms of what happens in biological systems. So we're very committed to continuing to find ways to do that.
Yeah, no, I think that's really fantastic, especially because there's such a good chance that really everything that happens on orbit ultimately can have an impact here on Earth, both in terms of automation. I think lab automation is something that probably most researchers dream of ultimately, and in most academic institutions is not necessarily a standard. But if those things become more and more common, I think it will elevate all of our science, because the more automated something is, the more we take the human factor out of it, right? I want to spend the last few minutes, actually, we're almost at the end, maybe picking your brains on the ST42, just so we can kind of dream of the future there, because I think it's a really cool project and the vision in that. So if you have a couple minutes to share on that, that would be fantastic.
Yeah, I'm going to defer to Jana since she's been with Space Tango longer. I think she knows a little bit more about the intricacies of the plan.
Yeah.
I'll be part of the, hopefully part of the implementation of the process here at Kennedy, where we hope to launch that from in the future.
Yeah, well, and Christine, it's really interesting for me because, when I first joined the space industry almost 6 years ago now, there was a lot of talk about how we were going to cure all these diseases and help people on Earth. And, just always just kind of chuckle a little bit and sort of say, well, that's great and everything, but you really couldn't do that on the International Space Station, because clearly to produce products that you can actually use in people, you need to produce them under some pretty strict manufacturing controls. We have obviously the FDA, our Food and Drug Administration, who approves drugs for safety and efficacy. The manufacturing piece of the approval is one of the biggest parts of that process. And so ISS is pretty constrained in terms of space to begin with, physical space. You probably saw in some of those pictures that Bill showed earlier in his presentation, every wall is covered, floor, ceiling, sidewalls, there's no up-down orientation. So space logistically is a little constrained. And it's also a facility that's been inhabited for the last 20 years by people. So not the cleanest in terms of where you might want to be manufacturing. And the other, logistical challenge is that if you installed a GMP facility on ISS, even if you had the space and you could actually, approach the levels of air handling systems that you would need to keep it in that kind of clean state, to inspect it would require FDA inspectors to go up to ISS, right? And I know a few cool FDAers who are probably willing to suit up and go up there, but the practicality of it is a little bit challenging, at least right now. I mean, we're increasing our access and that's fantastic, but it's not like you could just jump on a flight tomorrow and go up and take a look if you needed to. So when I first met Twyman, our CEO at Space Tango, he sort of had this idea of ST42, which is this automated capsule that has no people on it, so it's not rated for human spaceflight and doesn't dock to any kind of a space station. So it basically gives you the ability to have that facility that you could build, that automated facility on the ground, send it to microgravity so that you could have the benefits that you see in microgravity from a manufacturing perspective, and then bring it back down so that you could return products that would be produced on orbit. And, you know, I loved the concept when I saw it, because I thought it breaks the chain and sort of takes that barrier out that we have right now. And I think the idea of manufacturing GMPs is something that is really new to the space community, but the idea of opening that whole market by laying that foundation with those capabilities is really important. So ST2 is our view of how on-orbit manufacturing, especially for products that are intended for human use, potentially could be achieved. And in you know, we didn't talk a lot today about the technology side, but we do a lot on the technology side as well. And in some of those processes, volatile chemicals or things that you don't necessarily want people exposed to, that sort of free-flyer capsule idea on our own schedule, launching kind of when we need it to launch, staying on orbit as long as we need it to be there, and then bringing it back once the process is finished is an important piece in terms of this commercial market development.
No, that's really quite brilliant, especially the own terms of it. I think is really something that's going to be crucial to the customers eventually. Essentially, if it's about producing something and then not being reliant on, well, when is the resupply mission coming back, etc. So that's something. It's a really brilliant idea. I'm really looking forward to it. So can't wait for that too.
Us too. Yeah, the future is really exciting. I mean, I think that pace in the last 5 years of what's happened has been amazing, but I think the next decade we're going to see some pretty amazing things as well.
Yeah. That's fantastic. We have one last question from the audience here, and that is specific about experiments that focus on protein crystallization. How long does it usually take from providing the sample to have the sample back? I'm guessing this is specifically targeted towards flying it to the ISS and then returning it.
Well, and I'll take that one really quickly because, you know, so all missions to ISS, and I think Bill mentioned in his presentation earlier, you can get to ISS on 2 different vehicles in commercial resupply flights right now. So you can either go in a Northrop Grumman Cygnus capsule or you can go up in a Dragon capsule. If you go up in a Cygnus capsule, though, you're not returning on that vehicle because that is the trash vehicle. So it is filled with trash and burns up on reentry. So if you went up on a Cygnus, in a Cygnus capsule, you would have to wait for a Dragon capsule to come home. And so typically we like to go up and back in the same mission, which is the Dragon capsule. And mission time is about 30 to 45 days right now. Ready to extend the missions a little bit from 30 days. So we really don't have a rapid return system quite yet to bring something home sooner than that 30 days. Your experiment may finish in 10 days on orbit, but it has to wait for the vehicle to come back. Hopefully at some point, you know, there's a lot of talk about return vehicles and we may have that capability. And with the increasing number of flights that are going up, there are other vehicles that will be able to catch a ride on as well.
Mm-hmm. Mm-hmm. Fantastic. Well, thank you. Thank you both so very much for making the time today and for joining us. And as mentioned, this is all recorded and we're really happy to share this with, we have, I believe over 270 participants in the symposium at this point, and it will remain on our YouTube channel and we'll keep advertising for it. I think, you know, the capabilities that your company have developed are really incredible, and yeah, can't wait what the future brings. And thank you so much for joining us here today, and I hope you have a great day.
Thank you, Chris.
Thanks.
Thank you.
Bye-bye.
Thank you. And to the rest of the participants, in about an hour, feel free to join me once again, and I will be talking to Dr. Erika Wagner from Blue Origin, and we're going to have some really exciting conversation there as well on how to get to suborbital flights and how to get your science onto those rockets. Until then, thank you very much for joining, and goodbye.
Bye.